definition. And so what would be the best way forward? So here are some examples of some initiatives that, for example, differentiable physics engines can actually integrate the body into the deep learning pipeline. Surely we can look at how infants learn and how babies learn. Neuromorphic intelligence is a topic. This is an article on the timescales by Fumiya. So I think we should sort of reflect this and talk as a community what would be the best way forward, how to sort of not mentor them that they don't understand embodiment, but to sort of, that they embrace, all the knowledge that we have gathered over the years. And I'll stop here. Thanks.
Thank you very much, Matej. I think that's a wonderful starting point to open up many discussions. And as you say, to be slightly provocative about kind of this area and what we're exploring. Fantastic. So we'll come back to Matej shortly to lead the discussion. But our next speaker that I'm glad to invite is Mariana Medina Sanchez. So Mariana, thank you very much for joining us. If you want to share your screen. Hopefully that works okay. Hi. Thank you very much for joining us. Let's see if your screen share works okay. Fantastic. Thank you very much, Mariana. I'll hand over to you. Oh, it's on presenter mode at the moment.
Okay, just one moment for this.
Perfect. Thank you.
Now, first of all, thank you very much for your kind invitation to participate in this interesting meeting. And I would like to share some of our research activities and how we can understand embodiment intelligence in medical microrobotics and some of our applications in the field of gynecological healthcare. I'm coming from the institute, from the Leibniz Institute in Dresden, Germany, and I was recently appointed as a professor here in San Sebastian, Spain, with Signa Nogune. So just a brief introduction of my research group. We are mainly interested in designing and developing microrobots or micro tools that interface biological samples at the cellular and molecular level for both fundamental studies as well as healthcare relevant applications. And these devices range from small scale smart sensor actuator systems and medical microrobotics. So for people who is not familiar with this terminal or with this concept, a medical microrobot can be an anthether or tether device, which is in the size range below 1 millimeter and possess in a way, like we would talk about embodiment intelligent or physical intelligent, because these tiny structures have not enough, let's say, space for accommodating sophisticated electronics and actuator sensors, as we know in conventional robots. So we have to rely more on the physical intelligence and how to functionalize these structures to make them multifunctional, that they are able to detect their surrounding environment, as well as to adapt their motion, considering the complexity of the biological environment where they are operating in. And it's also important to note that the locomotion mechanisms at this scale are dominated mainly by viscous forces, and making this very challenging. No? And we have to get some inspiration from nature and use non-reciprocal motion mechanisms to be able to manipulate them in biological fluids that typically are quite viscoelastic and contain also several protein and molecular debris. So as I mentioned, these can be anthether, but also tether. So we can talk also about catheters that are in a way smart, because they can be fabricated with smart materials or with materials that respond to certain stimuli, like for example, these elastomers that are embedded with magnetic particles, so that by employing external magnetic fields, one can manipulate the position and function of these structures. So just a brief introduction of which kind of microrobots or microrobots we find in the literature. So they are basically classified by the locomotion principle, and the main ones are the chemical, physical, and biohybrids. The chemical ones employ chemical reactions, for example, enzymatic reactions, for example, in presence of biological fluids like glucose or urea. If you have an immobilized enzyme on these synthetic structures, they can produce certain chemical reactions that lead to gas production and propel the structure forward. They can be also physical, employing external magnetic fields, light, or ultrasound to propel them remotely. Biohybrids, which in combination to motile cells or microorganisms, they have interesting functionalities for biomedical applications because these microorganisms or cells are already adapted to swim in these complex environments. And they can also interact with biological materials, fuse with somatic cells. And the advantage of the engineer or the synthetic structure is that you can load different cargos, take advantage of the different surface chemistry, depending on the materials that are employed. They can also serve as contrast agents for imaging once operating them under scattering tissues. And I also want to highlight these other two because biological ones are obviously the inspiring kind of robots, or extractors, but also there is a lot of development on synthetic biology, and a lot of work is being done using genetically modified bacteria to also serve as vectors for example, cancer treatment or to assist immunotherapies in a more effective fashion. And on the other side, the purely electromechanical ones, which are getting closer to what a real robot is, where they are still not as small as the previous ones or single cell. They are more close to the millimeter size range, but they already include some actuators, energy receiver coils, and micro heaters that make these structures also more multifunctional. So the main application of these robots vehicles is the operation inside the living body. And there are various applications that people in the field are exploring, like, for example, tissue engineering, where these carriers have been, for example, loaded with stem cells and externally actuated by magnetic fields to the injury site to induce tissue regeneration. They have also been used for cancer treatment. For example, this is a magnetotactic bacteria that is decorated with drug-loaded liposomes. And these liposomes are interacting with the cancer cells locally, inducing cell apoptosis. They can also be used for blood clot removal and even molecular sampling, like, for example, using these molecular beacons with quantum dots that are whose light is or emission is quenched depending on the biomolecules that are in the surrounding fluid. In our particular case, as I mentioned at the beginning, we are mainly interested in the field of gynaecological healthcare and how we can use these microrobots for assisting fertilization in vivo, especially in cases of infertility, like male infertility, when sperm motility is quite low or sperm count is very low, but sperms are still able to move powerfully. So, and here we see some statistics, and actually conventional assisted reproduction techniques are quite successful nowadays. But in terms of fertilization rates, the numbers are quite high. The problem comes when the fertilized embryos or fertilized oocytes are transferred back to the uterus. Then is when we observe significant drops in the implantation rates between 17 to 21%, requiring multiple transfers. and also with the risk of having multiple births. So our vision is to create these structures which are at the same size scale as the gametes, meaning sperm cells, oocytes or early embryos, and use these microcarriers to bring them nearby the fertilization site in the fallopian tube non-invasively using external magnetic fields. But for that, we need also not only a proper geometry, but also a proper selection of materials that provide this physical intelligence, like, for example, the gripping function, that the gripper is able to open and close to release the cargo, or, for example, to react upon certain stimuli to release a therapeutical compound, or to induce the local activation of sperm cells once reaching the oocyte. So basically, the main goal is to perform accurate operations at the cellular level and reach very difficult locations in the body non-invasively. And this is how the setup would look like: a set of electromagnetic coils that will allow us to manipulate these tools remotely in 3 dimensions while imaging them in real time using modalities like ultrasound or photoacoustics. So here are some examples of this manipulation of cells. So one is the case of sperm cells that are in low count, but they are motile, so we functionalize them with magnetic microparticles. And here, using optical feedback algorithms, we can correct the trajectory of these sperm cells and move them towards the desired location. In the case of low sperm motility, which is another male infertility factor, what we propose here is the use of an artificial flagella, which is made by 2-photon lithography, made of a biodegradable polymer with a very thin film of ferromagnetic material. And then we can manipulate them to capture the immotile sperm cells, transport it to the oocyte, and release it. So coming back to the physical intelligence, so how we can go a step further, because these materials so far, the ones I showed in the previous slide, they are like rigid materials. Even though they are polymeric, they don't have a specific response just to the magnetic field that we are using for the transport of cells. But we need to provide additional intelligence and additional functionality, and this is achieved by the selection, by the proper selection of a material. And here we are taking advantage of hydrogels that respond to different stimuli. And in particular, we have been working with polynipam, that is a thermoresponsive polymer, whose lower critical solution temperature can be tuned, adding different compounds like acrylic acid to have a response close to the physiological temperature range. But we can also use other materials that respond to pH, humidity, or specific chemical reactions. And I just want to show you a couple of examples. This is a multifunctional carrier where we have an hydrogel, the polynipam hydrogel, within the sperm carrier. And this hydrogel is serving as a microvalve so that we can release sperm cells on demand in the right location. But at the same time, this hydrogel is loaded with heparin, which is used in vitro fertilization for capacitating the sperm cells to be able to fertilize the oocyte. Additionally, we have on the surface of these microcaps nanocarriers called polymersomes, which also have a particular surface engineering. Maybe here I'll show you. So these are nanocarriers that are loaded with an enzyme called hyaluronidase. And these polymersomes also are sensitive to a stimuli. In this case, it's a pH stimuli. And we selected these two stimuli, temperature and pH, because in the fallopian tube, in the reproductive tract, it's found that between the uterus and the fallopian tube, there is a gradient of temperature as well as a gradient of pH. So this would be the perfect, let's say, stimuli, not only to help capacitating these sperm cells in cases of infertility, but also to release these enzymes to facilitate the journey of the sperm cells through the biological barriers that surround the oocyte, like the cumulus cell complex that is like tightly bound by hyaluronic acid. So this enzyme is breaking these bonds and like loosening the path of the sperm cells to reach the oocyte more efficiently. So here, I think it's a clear example of how we use this physical intelligence for very complex tasks at the smaller scales. Yeah, another example is these sperms that are loaded with
Mariana. We're just going to have to wrap up in a few minutes if you can, and then we can come back to this in the panel discussion. Sorry to interrupt.
Yeah, so should I finish already, or I didn't understand?
Yes, sorry for that. If you could just finish off. Thank you very much.
Yeah, so actually this was my last slide. So this is another example of these reapers using the same material, and we can just with very small changes of temperature create like opening and closing of these structures. Okay, so maybe just to finalize, this is my last slide where we talk about medical microrobots and how we are like trying to make progress to bring these devices close to the clinical and clinical tests. And two of the main challenges is imaging and control. And yeah, maybe in another time I will talk to you about some ideas how to also provide other kind of functionalities and contrast of these structures to be able to visualize and control them in living animals. And with that, I would like to thank you for your attention. If you have any question.
Thank you so much, Mariana. So sorry to interrupt you. Really fascinating work. I think it's a really nice application of this physical intelligence, as you say. So we'll keep questions to the end, and we have the panel discussion. So hopefully, Mariana, you'll stay with us and join us back in a few minutes with Matej. So thank you once again, Mariana, for a really interesting, fascinating work. So